So now we're going to start talking about the maintenance of blood pressure. So this is the beginning of your part 2 PowerPoint slides for blood vessels. Okay, first of all, there is a difference if we're talking about short-term maintenance of blood pressure, if it just changes for a short amount of time, or long-term. So talking about short-term first. Mean arterial pressure, that is what MAP is, must constantly be maintained right around that 95 millimeters of mercury. So if that goes down or if that goes up, that's going to trigger the nervous system, the endocrine system, or the urinary system to do something to bring the body back in homeostasis, which involves keeping MAP at 95 millimeters of mercury. So blood pressure is equal to the heart rate times the stroke volume times the peripheral resistance. Keep in mind, heart rate and stroke volume multiplied together equal cardiac output. So short-term control of blood pressure is primarily able to be controlled by the nervous system and a few of the different hormones of the endocrine system. They will either adjust the resistance, increase or decrease resistance, or can change cardiac output. And we talked about those inotropic and chronotropic effects that do that with the heart in that chapter. So how the nervous system can help maintain blood pressure short term. So of course, with blood pressure, heart, smooth muscle, you're talking about the autonomic nervous system is what's going to be in control. And so both the divisions, the parasympathetic and the sympathetic, both have an immediate effect on blood pressure, if it will be lowered or raised. Obviously, parasympathetic, it's going to be lowered. Sympathetic, it will be raised. So sympathetic, how do they raise blood pressure? They release norepinephrine and norepinephrine onto the cardiac muscles and also in the smooth muscles of the blood vessels. So you have cardiac muscles contracting more, so you're increasing the contractility, which enables you to increase stroke volume. Also, epinephrine is going to increase heart rate. It increases the rate of the SA node. And going to the smooth muscle, it's going to cause stimulation, which is vasoconstriction. So you're increasing the peripheral resistance, which therefore increases pressure. So keep in mind that the heart, we have sympathetic axons that are going to go to the AV node as well as to the cardiac muscle itself. Okay, we also have it going to the blood vessels. So anytime basically your core, your hypothalamus decides that you're in sympathetic, you're going to carry out these responses. Which sympathetic response, we want to raise blood pressure because we want more blood to be able to go to more tissues to allow this increased body activity. Parasympathetic system, and the main nerve is the vagus nerve. We're going to release acetylcholine, or ACH, onto cardiac muscle cells at the pacemaker. Remember, ACH really doesn't have an effect on cardiac muscle itself. It does also affect the AV node. So it's going to slow down the rhythm of these. So it decreases heart rate, which decreases the blood pressure, which obviously decreases cardiac output. Parasympathetic doesn't specifically do anything to blood vessels. So because nothing's done, we are not stimulating it, naturally it's inhibited and the blood vessels will be more dilated. So peripheral resistance is decreased, blood pressure is decreased. So when blood pressure increases above the normal range, okay, so basically that's going to cause your 95 millimeters of mercury that you're supposed to have with your MAP. We're saying that gets too high. You have receptors that are measuring pressure in your aorta and in the common carotid arteries. So they measure and sense that the pressure has increased. they immediately are going to send information to the control center. So this is the medulla of the brain. Remember, the medulla oblongata actually has nuclei that control the heart, the cardiac center that control heart and blood vessels. So parasympathetic neurons will decrease activity of the intrinsic conduction system. So they slow heart rate by affecting the SA node. And you have the inhibiting of smooth muscle, so the vessels will be dilated. Both of those things are going to decrease blood pressure. So of course, this is an example of a negative feedback. So when blood pressure is too low, same place, the receptors are in the aorta, common carotid. They sense the pressure is low. They send the information to the same part, your cardiac center, and the medulla oblongata. So now we are going to, with sympathetic control, that's going to increase activity of the SA node, increase cardiac muscle contractility, both of which increase cardiac output, and it causes contraction of smooth muscle around blood vessels, which increases peripheral resistance and also increases blood pressure. You still, I know that's a lot to kind of grasp, so if you would like to watch a video, it's very helpful in showing how the baroreceptors respond with the different stimuli. Valsalva maneuver is used to test the baroreflex and can actually be used if you want to increase the heart rate. So the subject will try to exhale against a closed glottis, which is in the voice box, which we call a larynx. This is what happens when we cough. So this is going to increase the pressure in the thoracic cavity, which puts pressure on the blood vessels, therefore decreases the amount of blood that can return to the heart. So if you decrease venous return, you're going to decrease cardiac output, and that will decrease your blood pressure. Naturally, if blood pressure gets lower, remember than the map, the 95, your body's going to do things to try to increase it. So that's naturally going to increase the heart rate to try to increase the blood pressure. Okay, now talking about chemoreceptors. So again, chemoreceptors are sensitive to chemical changes in the blood. The locations of these are the same place that we have baroreceptors, so in the walls of the aorta and the carotid arteries. Peripheral chemoreceptors play a role in regulating breathing. They also can affect blood pressure. They're most sensitive to the levels of oxygen in the blood. Central chemoreceptors, these are in the brain, along the brainstem. These respond to a decrease in pH. If you decrease pH, that means you've increased the amount of hydrogen or the acid in the blood. This will trigger a feedback loop that indirectly increases the activity of sympathetic neurons. Sympathetic neurons naturally are going to cause vasoconstriction, and they're going to increase blood pressure. The endocrine system works with the nervous system. Of course, both are regulatory systems of the body. Although it is slower, like all hormones are slower than neural stimuli. So we've talked about these. So epinephrine, norepinephrine, of course, these are going to increase cardiac output. Thyroid hormone, an increase in that increases metabolic activity. So that's also going to increase cardiac output. So remember, epinephrine, norepinephrine are released from the adrenal medulla. They also are going to cause vasoconstriction. So they increase activity of the heart, which increases blood pressure. Vasoconstriction increases resistance, which also increases blood pressure. Another powerful vasoconstrictor is going to be angiotensin II. Remember, this is part of the Ross system. So when blood pressure is too low, the kidneys release renin. Renin makes us release just simply angiotensin I. gets converted to 2. It's actually 2, that stands for aldosterone. And that causes constriction, because if you're dehydrated, your blood volume, blood pressure going to be low. So that will help increase blood pressure as well. Of course, ANP is going to do the opposite of what angiotensin 2 does. It decreases the peripheral resistance slightly because it's released when blood pressure is too high. So if you lower peripheral resistance, you will decrease the blood pressure. So this is basically a map that you should be able to work yourself through. Again, this is short-term effects, how we regulate blood pressure. So here we're looking at mean arterial blood pressure, which of course we calculate with stroke volume times the heart rate, of course that's our cardiac output, times peripheral resistance. So we know, as we already talked about, the adrenalines or catecholamines, epinephrine and norepinephrine, increase heart rate, which is going to help increase the blood pressure. We also know with vasoconstriction that they're going to increase peripheral resistance with vasoconstriction, which would also increase your mean arterial pressure. How do we know to release this? That's because of the baroreceptors, or there's receptors that are monitoring these changes in the stimuli. So they will activate the central nervous system, which initiates the autonomic nervous system, which causes the release of epinephrine and norepinephrine. Also, what's in constant communication with the nervous system are chemoreceptors. So in situations of hypoxia, low amounts of oxygen, acidosis, so high amounts of hydrogen or low pH, and hypercapnia, which is when you have a high amount of CO2. Typically, if CO2 is high, then oxygen will be low. So those go together. And when we talk more about how pH is regulated through the body, acid-base balance, we're going to talk about that CO2, the more CO2 you have, the more acid. So those will stimulate the chemoreceptors that will stimulate the same things we talked about from the baroreceptors. And then we know that chronic stress and the release of cortisol is going to overstimulate the central nervous system, stimulating the sympathetic to increase mean arterial pressure as well. Now we're going to discuss long-term control over blood pressure. So this is primarily urinary system, takes a lot longer to act, and other endocrine hormones that act on the kidneys. So we can control blood pressure by increasing or decreasing the amount of water that we keep on the body. We can control this by increasing or decreasing how much we urinate. Remember, blood volume has a direct relationship with blood pressure. So the endocrine system can help regulate blood volume through 2 different primary systems. So ANP, just this should be a review. When blood pressure increases, cells in the right atrium release the atrial natriuretic peptide. So this is going to be when pressure is too high. So this is going to make the kidneys lose more water. Also, they're going to decrease the amount of sodium. Both of those are going to increase the amount of urine, so decrease blood volume and therefore decrease blood pressure. Should be a review as well. So when blood pressure is too low, the hypothalamus can release ADH. This is going to make you thirsty. It's going to trigger the kidneys and make you reabsorb more water, both of which are going to raise blood pressure. Also, the kidneys have receptors for blood pressure. So when blood pressure is too low, they will release renin. Through that ROS system, you will have angiotensin 2 that's produced. Again, that will cause vasoconstriction. That will also make you release ADH, which, as we already said, makes you thirsty. It makes you retain more water. Also, it's going to cause you to release aldosterone, which is going to increase the amount of sodium that we're retaining. Both of those increase the amount of water that the body has and therefore increases blood volume and blood pressure. So if blood pressure increases, to describe this action, if blood pressure gets too high, so this is represented by our blood, we are going to move water and salt into the tubules of the kidney. Eventually, what is in the tubules of the kidney will become urine. So we're able to decrease the amount of blood volume, therefore decrease the pressure. We call this pressure diuresis. Okay, the force of urinating more to help control blood pressure. Obviously, this is the opposite. So if pressure decreases, we want to take the water that we would be urinating out and we want to reabsorb it back into the bloodstream. If we increase the blood, you've increased the blood pressure. So essentially, this is the same kind of chart, but now again, we're looking at long term. So the biggest difference long term involves the kidneys. So you should be able to talk yourself through these steps. You know, even things we talked about, hematocrit, red blood cells, EPO, all of these things that we talked about in the previous exam, as they relate to blood volume and blood pressure, you will be tested over those again. So make sure that you understand how all of these different hormones affect our mean arterial blood pressure. This is nice because this puts it together. So it's showing short term, of course, is everything on this side. Long term is on the other. Before the exam, you must be able to talk through this. Hoping that we actually have a little bit of time before the next exam so we can kind of go through this together as a class. I think that would be helpful. That is a lot to take in. So there are two really great videos that will help you with that. So I strongly recommend that you watch those. So to summarize how we maintain blood pressure, we can do it by 3 different systems. By the 2 systems that help regulate and control everything else, nervous and endocrine, and by the urinary, because we're really able to control the amount of water in the body, therefore pressure. So each one of these can impact the amount of blood that we have in the body, the peripheral resistance, or the cardiac output. These again are the 3 factors that affect blood pressure. So if you can affect either one of those 3 factors, you can affect blood pressure. This is just it's another way to look at it. So it's charts that individually show the different responses. So if you feel like this is the easier way for you to learn it, of course you can look at those. We already discussed. You can read over the carotid sinus massage. Basically, by putting pressure there, you're on top of those arteries. You're stimulating the baroreceptors, telling the pressure that it's too high, so it can help reduce blood pressure. We'll talk a little bit about the fact that you never want to do both carotid arteries at the same time in lab as well. Okay, so when blood pressure is not normal, again, it needs to be maintained at about the MAP at 95 millimeters per mercury. So if blood pressure gets too high, that value goes up. We term that hypertension, or if it falls too low, of course, we call that hypotension. There are several levels to hypertension. Anything technically over 120, you're considered, actually 120 to 139 is pre-hypertension. So anything 140 or higher, you will be diagnosed with hypertension. 2 basic types, essential or primary. They're not sure of the cause. They don't know why the blood pressure is, chronically high. Secondary, the cause can be determined either genetically or because of obesity or possibly, another problem that a patient's having. Hypotension, lower than normal blood pressure. We term it that when the systolic is lower than 90 or the diastolic, or both, is lower than 60 millimeters of mercury. And typically this isn't a problem. There are some people out there that have naturally hypotension. It's really only a concern when you show signs and symptoms, when the body is unable to adequately give oxygen and nutrients to the tissues of the body. So when hypotension gets severe, very, very low blood pressure, circulatory shock will occur. Basically, there's not enough blood to get to any of the organs. Organs start to fail, including the brain. So that's why you can lose consciousness, because there's not enough oxygen. Okay, one of the most common causes when it's this severe is because the person's going through hypovolemic shock. So that occurs when you lose a lot of blood volume. If you're bleeding internally, have a wound, if you have severe diarrhea and vomiting, if you've used way too many diuretics that would pull too much water off you, or if you're severely dehydrated and have not drank enough. Any of these can lead to that hypovolemic shock, which is fatal unless you're able to restore the volume of blood. So you can give the person, of course, blood transfusion or simply an IV bag. A decrease in heart rate can also lead to hypotension. So maybe it's not a loss of blood, or fluid, maybe it's something wrong with the heart. So if the heart cannot stay at its normal pace. This is not very normal because the SA node sets the rhythm of the heart. So typically when this happens, someone's on a medication that is working too good. Okay, they have hypertension and it's lowering it too much. If the heart can't pump adequately, the stroke volume, the amount of blood ejected per beat, will go down. And so as stroke volume goes down, cardiac output goes down. So the heart tries to work harder to increase cardiac output. And so it starts to increase the rate of contraction. And eventually the heart fails because it is ineffective at being able to serve the tissues with the proper amounts of oxygen. And so the heart itself is not even getting enough oxygen itself. So this can cause cardiogenic shock. Excessive vasodilation can also cause hypotension. Of course, vasodilation is going to decrease peripheral resistance. When you decrease resistance, you're decreasing blood pressure. Again, this would have to be excessive. So things that would cause this again would be medications for hypertension that are working too good. If the ANS system is not operating properly, or if blood pH is very acidic. Another potential cause is too much release of histamine, which occurs when a person's having an allergic reaction. So if someone's deadly allergic to bees, that's why it can be deadly, because it leads to anaphylactic shock. Also, if the body has had a lot of trauma, a really horrible car accident where there's a lot of trauma everywhere, remember histamine is going to be released before tissue repair, and that could actually lead to shock as well. Excessive vasodilation, if that occurs because of a bacterial infection in the blood, then we call that septic shock. So the signs and symptoms to all types of shock would be low blood pressure. The person's going to feel fatigued, tired. They might be confused. They might lose consciousness. It's all going to be the same. You might even have an increase in heart rate trying to overcome this, because in all situations, the blood pressure is too low. So your nervous system, endocrine system, and urinary system are all going to try to bring blood pressure back to the proper homeostatic range. So a little review. Which of the following is not true? The cavities in the temporal bone are called carotid sinuses. No, they're within the carotid vessels, so that's obviously false. The carotid sinus reflex regulates blood flow to the brain. That is true. This is a reflex that constantly regulates how much the blood pressure going to and from the brain. That's why we have chemoreceptors and baroreceptors in the carotid arteries and not just in the aorta. If you stimulate baroreceptors, pressure receptors in the aorta, that means that pressure would be too high, so that would help lower and normalize the blood pressure. That's true as well. If you stand up very quickly, that will reduce the flow of blood to the brain. That's true. That's why sometimes you might feel a little dizzy, because as you stand up abruptly, the blood pressure is changing and the body has to adjust to that. Baroreceptors sense changes in vessel stretch. That is true. The higher the pressure, the more the stretch. Chemoreceptors are associated with detecting hypercapnia, which remember is too much CO2, which lowers the pH, causing it to too acidic. That's what acidosis is. Chemoreceptors do detect when oxygen levels are low. That's what hypoxia is. They are found surrounding your common carotid arteries. And they do provide input from the respiratory center. Okay, if oxygen levels are too low or CO2 is too high, they want to change your respiratory rate as well. So located near proprioceptors, that gives you information about the position of the body. It has nothing to do with that. Which of the following is not one of the negative feedback systems that responds to hypovolemic shock? So remember, hypovolemic shock is when you've lost a lot of blood volume. So your blood volume is low, therefore blood pressure. So to increase blood pressure, we would release or increase the amount of epinephrine and norepinephrine that are released. We would increase the amount of aldosterone that's going to be released from our adrenal cortex. That will help increase blood volume because it increases sodium retention. We're going to increase the amount of ADH, increasing water retention. And both the hormone that actually will trigger the release of aldosterone and ADH is renin from the kidneys. Remember, they release renin when blood volume is too low. So the one that doesn't work are release of local vasoconstrictors. Okay, which type of shock is caused by an inappropriate dilation of small vessels? So anaphylactic shock is when you have a severe allergy, and that's because of the release of histamine, so that would be true. Hypovolemic, you're losing the amount of blood, so that doesn't have to do with dilation. Obstructive would be from plaque buildup or possibly a blood clot, a thrombus. Septic shock occurs during a bacterial infection, which would cause extreme dilation of blood vessels. So the correct answer is anaphylactic and septic shock. So we briefly discussed capillaries when we were discussing vessels, arteries, and veins, but now we're going to get more at the physiology, which is tissue perfusion than capillaries. So as we mentioned earlier previously, capillaries are found in clusters, several vessels together. And they're going to be around the cells between typically an artery and a vein. So the way that blood moves, flows to a tissue through the capillary bed, we call that tissue perfusion. So if tissue perfusion is what it should be, then that means your cells are getting and tissues are getting the adequate amount of oxygen and nutrients to them. So as we mentioned previously, capillaries are very thin vessels, extremely thin walls. They basically only have that internal layer, the endothelium, which are the simple squamous cells, and the basement membrane or basal surface that they attach to. So this is one of the things that allows for easy movement of substances in and out of capillaries. Because they're so thin-walled, we want to make sure that they don't collapse. So they have cells that surround them called pericytes that help hold capillaries, support them. and therefore help direct and control blood flow. Remember from AMP1, all epithelial cells have tight junctions between them that's just going to help hinder flow of anything between the cells. So this is what I would call a normal capillary bed. Okay, very, very small spaces. Again, the endothelial cell makes up the bulk. And again, some capillaries are so small, as we see here, only one red blood cell can fit at a time. Capillary exchange then is going to be the movement of nutrients, gases, ions, waste, anything that you want to take to the cells or remove from the cells that travels between the capillary and the tissues. So the movement of these materials, we call that the capillary exchange. And there's 3 different types of mechanisms that capillaries can exchange this material. 2 types of diffusion, so those are going to be passive processes, and then transcytosis, which is an example of an active process. Water moves primarily by filtration here. Filtration is driven by pressure, not concentration. So again, the higher the pressure, if you have high pressure, low pressure, naturally we're going to move from high to low. And we'll talk about that a little bit more. Some capillaries will actually have small pores or holes within their cells. We call these fenestrations, small little gaps. So diffusion and osmosis can occur through these small gaps or pores. Water is relatively a small substance as well. So any substances that can dissolve in water can easily move through these pores as well, as long as a gradient is present. So you're always going to go from high to low. Lipid-soluble substances like gases and very small lipids can directly diffuse straight across the phospholipid bilayer, leaving the capillary bed and entering the interstitial fluid, and then it can enter inside the cell. Or if these are things that are moving from the cell back into the capillary bed, it can move that way as well. Again, what's going to direct the movement of these lipid-soluble substances will be the gradient. Transcytosis, this is a form of active transport. Very large molecules have to basically form a vesicle and move across the cell. So molecules first are taken into the cell by endocytosis, and then they're going to be removed by exo. So we're transporting it across. That is what transcytosis is. So diffusion and transcytosis involve a substance that has to cross those endothelial cells. Okay, so to recap here, things that are lipid soluble can move across those blood cells. So of course, if it's moving away from the capillary into the interstitial fluid, or it can move from the interstitial fluid back into the blood. That's passive, so that's going to be along the concentration gradient. Also passive is going to be diffusion and osmosis, which occurs between these natural little gaps that we have present between endothelial cells. And then in this case, you have those small pores, your little fenestra. Again, you're going to see the movement of particles from high to low concentration. And then, of course, transcytosis. Large stuff has to be packaged in a vesicle. So we bring it inside, endo, move it across, and release it. So there are actually 3 different types of capillaries. In certain types of tissue, you need a different type of capillary bed. So we will briefly compare and contrast the 3 different types. So continuous capillaries are the most common that we have. Pretty much if it's not listed as one of the other types, which are fenestrated or sinusoidal, assume that it's going to be continuous. So these endothelial cells have tight junctions, so it's hard for things to pass between cells. So we have this in the skin. That's part of what makes skin a protective barrier. most nervous and connective tissue, and muscle tissue. So again, almost everywhere in the body. As far as how easy is it for things to leave the capillary or enter, these are going to be the hardest. We refer to these as the least leaky. Okay, so they're very selective in the substances that can enter the blood vessel or exit. Slightly more permeable are the fenestrated capillaries. So they actually have these small little tiny pores that allow additional passage of substances. So think about where in the body are we constantly going to be moving stuff in and out of the blood. The kidneys, as we've already been talking about, they help us regulate blood glucose levels, several ion levels, water. So we're constantly moving stuff. If you want to keep it in the body, we're going to move it back into the blood reabsorbed. If we want it to be in the urine, we're going to make it exit. So we want that process to be relatively easy. Endocrine glands do not have ducts, so we need hormones to be able to easily enter the bloodstream to get to their target. And then in the small intestines where we absorb nutrients, we want that to be easier. The most permeable capillaries are what we call sinusoidal capillaries. So you have very large spaces between the cells, again, making it very permeable, very easy for stuff to move in and out. So they're what we call the leakiest. They will allow the largest substances to be able to move across. So we have this in the liver. You have a lot of large proteins, fats, sugars. Again, we're constantly moving things in and out of the liver based on the metabolic regulation of the blood. Lymphoid organs, we want it to be large enough for white blood cells to easily move in and out of the bloodstream. Same thing for bone marrow. And spleen is where we clean and remove old damaged red blood cells, and we also kind of check there as part of lymphatic function as well. So all of those areas you want the most permeable capillaries, which would be the sinusoidal. So the flow into a capillary bed, we call that microcirculation because how small the actual diameter of most capillary beds. So this involves two different types of vessels. True capillaries, where materials are exchanged, and then a central vessel that basically just carries blood from the arterial end to the venule end of the capillary bed. We talked about this very briefly in class today. So actually, I kind of just want to go to the picture. I do want to mention at rest in parasympathetic mode, okay, only about 25% of the body's capillary beds are fully open. Okay, so a lot of that blood is going to be in our veins as the blood reservoir for when we need it. Remember, most of the tissues are resting. They don't need the extra nutrients and the oxygen. Okay, so as we previously discussed, this would be our arterial end, so blood's coming at a higher pressure on this portion of the artery. Of course, the capillary bed's going to be drained by small veins, which we call venules. So this, and I talked about this meta-arterial can go straight to the post-capillary venule. That's that one thoroughfare channel. So that's still allowing some blood to enter a particular tissue. So true capillary bed, if it's actually all open, then you would also have blood moving between all these little vessels that surround it. So to allow that to happen, these precapillary sphincters, which are bands of smooth muscle, will relax, allowing blood to enter the other portions. If you don't need a lot of blood in that particular area, like skeletal muscle when you're in parasympathetic, then these precapillary sphincters will contract, not allowing blood to enter these other portions. So you'll keep just enough blood keeping the tissues alive. Talked about all this. Really important to make sure blood does not backflow into true capillary beds, because on the arterial end, you're going to have a higher pressure, on the venule end lower. Remember, lower. Blood naturally moves from high to low pressure. This is a great video if you would like to review when blood is just going through the thoroughfare channel, when the precapillary sphincters are contracted, or when they're relaxed and blood is allowed to flow through all the portions of the capillary bed.